Thank you very much for having me for this seminar series, and I'm excited to share our recent work. And dopamine is involved in multiple functions such as reward-based learning, motivation, and movement. But how these functions are regulated by dopamine remains to be clarified. One of the influential ideas is dopamine signals reward prediction errors and regulate reward-based learning. The role of dopamine in reward has been indicated by intracranial self-stimulation experiments. Furthermore, Wolfram Schultz and colleagues have shown that the activity of dopamine neurons resemble so-called reward prediction errors or a teaching signal used in machine learning algorithms. It has long been assumed that dopamine neurons signal reward prediction errors to a wide area of the brain, including the striatum, which is the area receives the densest projection of dopamine neurons. The striatum is, however, not a unitary structure. It is thought that different regions of the striatum regulate distinct types of behavior. The striatum can be parceled into several regions, most commonly classified into ventral, dorsomedial, dorsolateral striatum. And this study, based on patterns of cortical inputs, showed that there is a fourth area of the striatum called the tail of the striatum. The ventral striatum is typically associated with Pavlovian behavior, and dorsomedial striatum, goal-directed behavior, and dorsolateral striatum, habit or skill. And all of these distinct behaviors are shaped by reward punishment, but whether these behaviors are shaped by the same prediction error signal remains to be clarified. Perhaps in line with diverse functions of dopamine, there has been increasing evidence indicating the diversity of dopamine signals. In addition to canonical prediction error signal, recent studies have found some movement-related activities, some increase or decrease near the onset of movement, or salience-related signals, or slow timescale signals, often called ramping dopamine signals. The challenge in the field now is whether we can understand these signals in a coherent theory or theories. Today, I will discuss the diverse dopamine signals based on our recent studies. First, I will discuss the idea of multiple dopamine systems or multiple axes for reinforcement learning. I will then talk a little about basics of reinforcement learning theory or temporal difference learning theory. And this serves as an introduction to the next topic, which is dopamine as moment-by-moment changes in value. And I will then briefly discuss a novel theoretical framework called distributional reinforcement learning. And finally, I will discuss our current overarching hypothesis on dopamine. Let me start by describing a very unique subset of dopamine neurons signaling threat. Many recent studies have indicated that dopamine signals varies depending on their projection targets. And those projecting to the ventral striatum, which originates in the VTA or ventral tegmental area, can be considered a canonical dopamine system. They are activated by reward or sometimes safety, and their signals are consistent with reward prediction errors. And optogenetic activation of these dopamine neurons cause positive reinforcement. We have recently shown that a specific population of dopamine neurons projecting to the tail of the striatum or the very posterior part of the striatum have completely different properties. And these neurons are located in the very lateral part of substantia nigra called substantia nigra pars lateralis. And they are activated by unpleasant air puff, loud sound, or novelty. And importantly, they do not respond to bitter taste, indicating that these dopamine neurons do not respond to all of negative events. And importantly, Optogenetic activation of these neurons caused opposite effect than the above, causing animals to learn to avoid such stimulation. These results together indicated that TS projecting dopamine neurons send very distinct teaching signals, suggesting that these populations define distinct reinforcement learning signals and forming multiple axes for reinforcement learning. So these dopamine neurons projecting to the ventral striatum send reward prediction error. Those projecting to the posterior part of the striatum send threat prediction error. What about other regions of the striatum, such as those medial striatum or those lateral striatum? We addressed this question in a recent study. And in this study, we used the decision-making task. And in this task, mice studied a trial by poking into an odor pot. We used odor mixtures containing two odors with different ratios. Mice were rewarded by poking into the left or right reward pot depending on the dominant odor in a mixture. In addition to varying the stimulus, we also manipulated the size of reward in order to facilitate our ability to monitor prediction error-related signals, as you will see later. In the first block of trials, the amount of reward was equal between the two sides. In the second block, however, the amount of reward was biased, with one side delivering big or small, medium reward, and another side delivering medium or small reward. And these two sizes at each location was chosen randomly with the probability of 50%. And this is a psychometric curve showing the mouse's performance. As you can see, in the second block, the performance was biased toward the side associated with big reward, indicating that the mice used both reward information as well as the odor information to make a decision. We monitor the activity of dopamine neurons at their projection site. We use calcium indicator GCAMP expressed in dopamine neurons and monitor the calcium signals in these three regions of the striatum, ventral, those medial, and those lateral striatum. And this is the average of all recordings, showing some responses to odors and during choice and reward. When compared between three regions, they showed relatively similar responses with some interesting differences. And first of all, the reward response contained similar patterns indicative of reward prediction errors. And their responses were modulated by size of received reward, small, medium, big, so small, medium, and big in all of these areas. And their response to the medium reward was modulated by overall expectation, meaning that the medium reward in the big side caused smaller response compared to the medium reward received in the small side. And this difference indicates the effect of expectation of reward, because in these trials, the same amount of reward was received. And comparing between three regions, there were some interesting differences. And the level of reward responses was overall positively biased in DLS. That is, a small reward typically caused negative response in ventral striatum and those medial striatum, but such negative response was lacking in those lateral striatum. So even the small reward did not cause negative response in the DLS. So overall, this reward response was positively shifted in those lateral striata. This overall positively biased response was also evident in other types of trials, such as error trials or Q predicting no reward. you can see in the ventral striatum or dorsal-medial striatum across different animals, you see negative response when the animal made an error. Whereas that negative response is missing in the dorsolateral striatum. And the Q predicting no reward also caused inhibition in ventral striatum and dorsal-medial striatum, but not in the dorsolateral striatum. So overall, the dorsolateral striatum did not show negative responses. So this study showed that the dopamine neurons in the 3 regions of the striatum are largely consistent with reward prediction errors. However, dopamine signals in the DLS, dorsolateral striatum, were positively biased. We argued in this paper that this biased response nicely explains the proposed function of DLS, which is habit or skill learning. First, a hallmark of habit is insensitivity to devaluation or the lack of reward. The diminished negative response might explain this property. The second, if the responses are positively biased, the effect of reinforcement would accumulate over repeated performance rather than balancing out prediction errors to converge on an appropriate value. The importance of repetition is highlighted by Thorndike's law of exercise. And we propose that DLS dopamine signals may be a neural instantiation of the law of exercise. And finally, DLS receives input from motor and somatosensory cortex. And this means that the DLS is well positioned to monitor motor commands and current body state, such as kinematics and posture, and drive next movements based on these bodily states. This would mean that elements of movement can be stitched together to form a sequence of action. And this may explain another hallmark of habit, which is automaticity. And overall, our study suggests that each region receives unique teaching signals from dopamine, which may result in different types of behavior, such as habit and skill, and suggesting that these areas concurrently learn different types of behavior based on unique dopamine signals and other inputs. Now let me talk about a different type of dopamine signals called ramping dopamine signals. Before explaining this work, I would like to introduce the essence of temporal difference learning algorithms. In this seminal paper by Schultz, Diane, and Montagu, they pointed out that the activity of dopamine neurons resembles a specific type of reward prediction error called temporal difference error, which can be formulated by this equation. Before discussing the next study, I'd like to explain what TD error is and why it is so powerful in machine learning first. And over the last 5 to 10 years, the field of artificial intelligence has made remarkable developments. In many of these applications, temporal difference learning or its variant has been used. And to understand how temporal difference learning algorithm works, let's take a look at a simulation. And in this simulation, a rat navigates in a maze to obtain a cheese. The goal is to find the shortest path to the reward. And what we first do is to define the structure of reward. The final location has a big reward plus 20 points. To encourage taking a shorter path, there is a small cost of taking each step, which is -1. And here's how the rat learns the maze. The color of each location indicates the value of each location. And as the rat moves around, the value of each location changes. If you look at it very closely, the color is changing around the location that the animal actually visited. And the color changes on the fly. So before even before reaching the final location, the color changes. That is, learning happens. And after learning this value representation, then the rat can follow this value representation to go to the higher value location. So critical feature of this learning algorithm is a local learning. And more importantly, values are updated on the fly, as you saw in the video, even before receiving a final outcome. And after taking one step, for example, starting from t to t+1, it computes the temporal difference error using this equation, which boils down to the difference between the value between two consecutive time points, v of t+1 and v of t. When TD error is detected, which means when there's a gap between values between these two consecutive time points, then the value of the previous state gets updated. And after each move, the value is updated locally, and after making many steps, it can build a smooth map of value across space. Let's look at what these equations really mean a little more closely. In reinforcement learning, the value of a given state or state value is defined as the discounted sum of all future rewards. The animal transitions across different states here, 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 at time t, t+1, and t+2. And at these transitions, animal receives different reward. And v of t is the sum of all of these rewards, starting from the current time point. And these rewards are multiplied by discounting factor gamma to emphasize more recent reward and discount future rewards. And observing this equation, this part is exactly gamma times v of t+1. So this equation can be simplified as the following. And this equation should be hold true if the prediction is accurate. But if the current prediction is not accurate, still being learned, this equation might not hold true. And the difference between the right-hand side and left-hand side can be considered as the inconsistency or error in the predictions. And this is actually how the temporal difference error is defined. So the difference between right-hand side and left-hand side is the definition of temporal difference error. And it is called temporal difference error because it uses the difference of values between consecutive time points. And this is used to update the value of the previous state. Now, one of the difficulties in reinforcement learning problems stems from the fact that reward typically comes after taking many actions. This raises the question of how one can distribute credit for success among the many decisions involved. And this is often called a temporal credit assignment problem. And TD learning solves this temporal credit assignment problem in unique ways. As we have seen in the simulation, this algorithm updates value even before receiving a final outcome. It did so based on learned values assigned to each location. And this means that the TD methods update their estimates based on their other estimates. In other words, they learn a guess from a guess because the value is still a tentative estimate, or they bootstrap. And why is this good for solving temporal credit assignment problem? Let's imagine two actions indicated by these arrows. If you have a good value estimates, one can tell that this action led to a better location, whereas this action led to a worse location. So even before receiving a reward, you can tell whether the action that you just took was a good one or bad one. And you can use this information to update your value. So from these arguments, it becomes clear that it is important that TD errors are computed on a moment-by-moment basis, even before receiving a final reward. And this is based on changes in value. And this is one of the reasons that makes this algorithm so powerful, so that it provides a solution to a credit assignment problem. Now, here is how TD errors can be applied to simple classical conditioning paradigm, where a Q predicts a reward. When the Q is presented, the value increases suddenly because the animal now knows that a reward is coming. The value goes down when the time passes beyond the time of expected reward, because now the animal does not know when the next reward is going to come. V of t plus 1 is just one time step shifted version of V of t. Because V of t plus 1 goes up earlier than V of t, taking the difference will result in a transient increase and decrease. Gamma is typically close to 1, so this is approximately the derivative of value. And when the animal receives a reward, this excitation can cancel this dip. Therefore, combining these three terms, you can get TD error, which approximate the dopamine response, explaining the Q response. So the response to the Q predicting reward, and diminished reward response when reward is expected, and dip in the activity when the predicted reward is omitted. And as discussed before, the TD error contains the term approximating the change in value or the derivative value. And the TD error explains all three features of dopamine responses in classical conditioning paradigm. So after introducing the TD error or TD learning algorithm, let me move on to the next topic, which is ramping dopamine signals. Dopamine ramps have been observed in multiple experiments, such as rats approaching and pressing a lever before obtaining a reward. Here, such as cocaine or sucrose. And dopamine ramps are observed also in navigational tasks using T-maze, or animals performing a sequence of actions to obtain reward. And dopamine ramps are observed in simple decision-making paradigm using nose poke as well. And most of these studies use cyclic voltammetry in the nuclear accumbens to measure dopamine concentrations. Yet, dopamine ramps are also observed in single-cell calcium imaging in the VTA. So after these findings, there have been hot debate about the nature of dopamine ramps. And some people argue that dopamine signals actually represent the value of the state rather than a reward prediction error. But considering the derivative-like property of TD errors, Sam Gershman pointed out that dopamine ramps can be TD error depending on the shape of value function. If the value function is discounted, if the value is discounted by a fixed factor gamma, and this term becomes 0. And that is when the value function is an exponential decay function using the discount factor gamma, computing reward prediction error or TD error results in 0 TD error. But what if the value function takes a different shape? For example, if the value function is more convex than the exponential decay function, computing TD error can result in positive or even ramping up TD error. And therefore, just by observing a ramping dopamine ramp, it is unclear whether we are observing a value function or TD error, which is a result of more convex value function. To dissociate these two possibilities, value and reward prediction error, we devised novel experimental paradigm. Imagine that a mouse is moving in a linear track to obtain a reward. What if the mouse is suddenly teleported to a new location? Let's assume that there is a monotonically increase in value function across space. When the mouse is teleported, mouse moves from here to here, and the value will increase suddenly. And this will result in a step-like increase in value. In contrast, teleport will cause a transient response in TD error or reward prediction error because it is the derivative of value function. Oops. Another important prediction is that the value should be always maximum at the goal location because there will be no more delay to reward at this point. And this does not have to be the case for TD error. And this manipulation therefore makes qualitatively distinct predictions for value and TD error. And another important illuminating experiment is a speed manipulation. If TD error is the temporal derivative of value, it should be sensitive to the speed of the movement. And fast speed will cause a greater ramp in TD error, while slow speed will cause a smaller response. The magnitude of ramp will depend on the speed. In contrast, the value should end at the same level regardless of the speed, because at the goal, now the animal is about to get the same reward with no more delay. It is important that this second experiment tests the property of ramp itself, whether it is more consistent with value or the derivative of it. We implemented these experiments using virtual reality in head-fixed mice. After animals were trained for a few days, they licked a water spout in anticipation of reward. And then to measure the activity of dopamine neurons, we used fiber photometry and measured calcium signals from the ventral striatum. In the first experiment, we included three conditions: long teleport, short teleport, and pausing at the same location. The value will show a step-like increase, whereas TD error will exhibit a transient response. The magnitude of these responses will be larger for long teleport than short teleport. When the animal pauses for a short period, the value may stay constant, whereas TD error may decrease back to the baseline since there is no change of value in time. Here is the result from 11 mice, and this is the average in the standard condition. Here is the response to a short teleport and a long teleport. As you can see, teleport caused transient responses, and the peak of the response well exceeded the peak of the standard condition. And this result directly violates the prediction of the value. A pause caused the ramp to disappear and the signal dropped back to the baseline. So these results are very much consistent with reward prediction error, but not to the value hypothesis. We next performed speed manipulations. Compared to the standard condition, the slow speed caused a weaker ramp, and the faster speed caused a greater ramp. And these results are difficult to explain by a value hypothesis. Instead, these results are consistent with the idea that the dopamine activity encodes TD error, or the temporal derivative of value function. We also performed experiments in which the speed was dynamically modulated, either starting from slow to fast or fast to slow. This makes very different predictions between value and reward prediction error. RPE will reflect the instantaneous speed, whereas the value reflects the position of the animal. And most notably, these two cases make very different predictions near the goal location, with two diverging for RPE and two converging for the value. And the result clearly supported RPE hypothesis, but not the value hypothesis. So the dopamine response really reflects the instantaneous speed of the movement. So one question that you may have at this point is whether the signals that we observed are just a sensory surprise. To address this question, we performed teleports between two different tracks. While running in one track, mice were teleported to another track occasionally. If dopamine neurons respond to a sensory surprise or sensory prediction error, we should see a response, but we did not observe such a response. As a control, we performed a forward teleport with the same animals, which evoked a big response, as in the previous results. We also characterized the single dopamine neuron responses in the VTA. We recorded spiking activity from optogenetically identified dopamine neurons. And these neurons showed characteristic responses consistent with RPE, consistent with the previous results using fiber photometry. And even at the single neuron level, you can clearly see the transient responses in response to teleport across most of the neurons. We have performed several analyses to characterize dopamine signals in a more data-driven manner. Here is one example. And this analysis seeks for a compact mathematical description of dopamine signals. In the field of mathematics, there is a method called fractional derivative, and this allows us to define the order of derivative, which is not an integer. The original function is the 0th order of derivative, whereas the most common derivative is the first order derivative. Using this method, one can change the function between the original function to the first derivative smoothly by changing the order of derivative. And using the photometry data, so in this analysis, we used some monotonically increasing value function and examined what order of derivative best explains data of dopamine signals. And using the photometry data, the best fit order of derivative was close to 1 across animals. Even based on single neuron dopamine data, the best fit order of the derivative was centered around 1, and very few were consistent with the 0th order, which corresponds to value and reward prediction error. So in summary, dopamine signals in the ventral striatum can be parsimoniously explained by the first order derivative of a function over space. So assuming some monotonically increasing function, dopamine signals can be explained by the first order derivative of that function. And dopamine signals are sensitive to changes in value, but not a sensory surprise. And these results indicate that these functions are consistent with what we conceive as value or TD errors, respectively. So even without starting from these concepts, we can describe the property of the dopamine signals as fast derivative of some monotonically increasing functions, which happen to correspond to a reward prediction error and value, as we usually discuss. And TD errors can manifest as slowly varying as well as phasic or transient responses. And in in total, dopamine neurons compute a derivative-like signals over value on a moment-by-moment basis, which is one of the fundamental properties of TD errors which makes the algorithm so powerful. And we also showed that the idea of moment-by-moment computations of TD errors applies to other task conditions, such as that I described before, this decision-making paradigm. And for instance, you might have noticed that dopamine signals dip even before reward feedback. So this is the time of reward. And this is when the animal enters a water port. And the dopamine signals dip even before receiving reward feedback. So another dip corresponds to the error, so reward omission. But another dip happens right after the animal enters the water port, even before receiving the reward feedback. So what does this dip represent? And this task can be modeled by different states and transitions between them, such as shown in this schematic. The animal receives an order and makes a choice, either big or small, and reward may be delivered or not delivered, and reward could be big, medium, or small. And these circles represent potential states in the task. To model this task, we added two different sources of noise, one for stimulus, adding perceptual uncertainty, as well as decision noise, which means that the animal does not always choose the best available option. In the presence of decision noise, something interesting happens. In this example, the animal receives a certain order, mixture, and estimate the current order as being here in this stimulus axis. And then due to the noise in the decision process, the animal happened to choose the small side instead of the big side. And this means that the animal lost a better option and chose a suboptimal option. What happens here is that the overall expected value decreases due to this suboptimal choice compared to the previous state. This causes the drop in the expected value, which can in turn cause a negative response in dopamine signals. In other words, we can explain this dip as a drop in the expected reward or the moment-by-moment change in the state value. In this paper, we showed that these dynamic responses not associated with cue or reward, also these discrete events, can also be explained as the moment-by-moment computations of TD errors in this decision-making task. So all three areas in the striatum exhibited dopamine signals largely consistent with reward prediction errors. And considering the task states, dopamine signals can be explained by moment changes in the expected future rewards consistent with temporal difference errors. And movement-related activity, contralateral orienting movements, caused phasic activation in DMS. So this is the movement-related signals that we observed in this data set, but not in other striatal regions like ventral striatum or DLS. And reward responses in DLS were positively biased and largely lacking negative responses even when the actual outcome was smaller than expected. And these differences in TD errors can differentially shape the behaviors controlled by each area. And we propose that these positively biased dopamine signals in DLS may correspond to Thorndike's law of exercise. Now, the studies discussed about using teleportation and speed manipulation provide strong evidence that dopamine neurons in the canonical dopamine system approximate TD error signals. Furthermore, those projecting to other parts of the striatum, DMS and DLS, contain modified TD error signals. What about other types of diversity in dopamine signals? In collaboration with people in DeepMind, we have recently provided some evidence that the canonical dopamine system contains the type of variability that can support a new reinforcement learning algorithm called distributional reinforcement learning. In conventional reinforcement learning, the agent learns to predict average future reward. So when the size of reward is varied across trials, probabilistically, it only learns to converge the average of future reward. In distributional reinforcement learning, instead the agent learns to represent the entire distribution of future reward. And this can be achieved by having dopamine neurons having different characteristics. And some dopamine neurons, such as shown in the red, respond more strongly to positive prediction error compared to negative prediction error. And the value predictors trained by these positively biased or optimistic dopamine neurons converge to the higher region of the distribution. In contrast, more negatively biased dopamine neurons, which is a more steeper response to negative reward prediction error and shallower response to positive prediction error, drives the value predictors to be down in the distribution. It converges to the lower region of the distribution. So if there is enough variability in the dopamine signals, then this population of dopamine neurons can together represent the shape of the distribution. In essence, the small variations in reward prediction error signals can help preserve detailed information about future outcomes. In this case, the probability distribution of future rewards. Combining these findings together, there are at least two ways the diversity of dopamine neurons can be interpreted. First, there are differences across target regions. The distinct dopamine signals can set up multiple global axes for reinforcement learning, training for different types of behavior. The second, even smaller variations within a subsystem can support distributional reinforcement learning so that more detailed information about future rewards can be learned and stored in the system. And this is an emerging functional view of the diversity of dopamine neurons, and we are currently testing these ideas using more experiments. With that, I would like to thank the people who are involved in the work. So the dopamine neurons in the decision-making task was led by Mitsuko Watabe Uchida and performed by Iku Tsutsui Kimura. And the tail of the striatum dopamine study was done by William Menegas together with Mitsuko Watabe Uchida. And the dopamine ramp was studied by Hyung-Gu Kim together with Athar Malik, Paul Beck, and theoretical aspects were contributed by Sam Gershman and his student John Michael. And we used the dopamine sensors developed by Yuron Lee. And the work on distributional reinforcement learning was done as a collaboration with a team in DeepMind, Matt Botovinik, Will Dabney, and Zeppert Nelson. And the funding comes from Brain Initiative and Simon's Foundation. Thank you very much for your attention.